So we've been talking quite a bit about chemistry in general, but in today's lecture we're actually going to apply the chemistry we've learned in earlier chapters and sections to the molecules of life. So essentially the macromolecules or biochemical molecules that make up all of life. And specifically we're going to focus on organizing these chemicals. I think this is one of the hardest tasks to do and one of the reasons why I'm going to focus on organizing the molecules of life in this lecture. So this is probably a review of an exercise we did in lab, but I just want to remind everybody that we eat life. So for something like this hamburger over here, we have plant products and we have meat products. So two major groups of organisms are represented by this hamburger. And if we look at the nutrition facts, so essentially some of the components of this hamburger, we can actually point out the major molecules of life. So fats are a major type of molecule that's found in life, carbohydrates, and proteins. So those are three of the four major molecules we find in life. And on this next slide, you can actually see all four. So we have lipids, carbohydrates, proteins, and nucleic acids. So these are the four major classes of biomolecules. Your lab also uses the word macromolecules, so biomolecules and macromolecules can be used interchangeably. And again, a major part of this lecture is trying to organize these four major groups of biomolecules. So here I've listed the common names for each of these. So for lipids, these are things like fats and cholesterols. Carbohydrates are things like sugar and fiber. Proteins don't have a common name, we just call them proteins. And you might know nucleic acids as being DNA and RNA. Again, going with this sort of organization theme, I've listed the three or four major macromolecules or biomolecules. And I've sort of split them up into different parts here. So for something like lipids, you have saturated versus unsaturated fats, you have cholesterols, and you have phospholipids. So those are the three major subgroups of lipids. For something like carbohydrates, you have simple sugars, you have cellulose, so indigestible plant sugars, and proteins, you have just amino acids. Nucleic acids, you have sugar, phosphate groups, and nitrogenous base. So here I'm just trying to help you organize your ideas a little bit, and this slide will make a lot more sense once we've gone through this lecture and you finish this chapter in its whole. So here's a question for you to answer on Blackboard. This is question number 1. So true or false? You are what you eat. So this is a little bit of a tricky question. So I'm going to leave this actually ungraded on Blackboard. But I'm leaning more towards the answer of A, or true. And so I want to remind you that you already are what you eat. So all of life is made up of the same four macromolecules. So this goes back to this concept of unity within diversity. So all of life uses the same four macromolecules. So this is a very good example of unity. There's also diversity in this fact. So some organisms can use different amounts and ratios of certain macromolecules. So for something like plant material that has a lot more dietary fiber or cellulose, compared to something like cholesterol, which plants usually don't have. So different organisms can use different amounts and ratios of macromolecules. Some organisms can also use these macromolecules for different functions. So we use sugar as energy and energy storage. Plants can use sugar for things like support. So again, going back to cellulose, that's the outside of a plant. So this plant body is essentially made up of cellulose, which gives it support. And some organisms can actually make small modifications of certain macromolecules. So we'll actually see in one of the questions coming up a modification to a sugar molecule. So as your lab manual points out, using the word macromolecule, you can think of these biochemical molecules or macromolecules or biomolecules, what have you, are all very, very large. So biomolecules are huge, huge chemical molecules. and they must be broken down or constructed in one way or another. So you can think of something like digestion. Digestion is all about breaking down huge biomolecules into something small that we can use. For something like building muscles, though, that's taking small, tiny pieces of biomolecules and building them up into something bigger. So in the case of muscles, that's taking small amino acids and building that up into proteins that make up our muscle. So the technical term for breaking down a macromolecule or biomolecule is hydrolysis. So this is a very important word. So this is taking polymers and breaking them down into monomers. So knowing your prefixes here is very helpful. So poly means many, mono means one. So it's taking a huge biomolecule and bringing it down into single pieces. So here we have a chain of 4 biomolecule pieces linked together into a polymer, and that's broken down into its monomer constituents. For constructing, so going from monomers to polymers, that's called condensation. Your book also uses the word dehydration. So these words are interchangeable, condensation or dehydration. And again, this is going from monomers to polymers. So here we have 4 tiny monomers of a biomolecule. That's taking them and linking them all together into one polymer. Again, monomer means one, polymer or poly means many. And so here's actually how those words get their name. So here we have a dehydration reaction on the left. We have a short polymer, so 3 purple circles. On the end we have a hydroxyl or OH group. On the other end we have just a hydrogen by itself. So through a dehydration reaction, water is produced. Notice this arrow points off into the distance. So this water is removed from the reaction, so it produces water. So that's where it gets its name, dehydration or condensation. So water is removed during this reaction. And as a result of a dehydration reaction, again, you're going from monomers to polymers, so you're building up. So here we've gone from 3 purple circles to 4 purple circles linked. Just as dehydration or building. On the right, we have hydrolysis. So the keywords here are hydro. You can think of water. Lysis is a word for breaking or destroying. So essentially this together means water breaking or destroying something. And that's exactly what this is. So here we have 4 purple circles linked together. 1, 2, 3, 4. Water, instead of leaving the reaction this time, actually comes in and helps the reaction proceed. So you have water comes in, and that breaks apart these purple circles. So we started with 4 purple circles linked together, and now we've broken off one of those purple circles. So now our polymer is only 3 monomers long. Here on this slide, we have some of the major chemical groups found in organic compounds. So I should really quickly define what I mean by organic. So these are carbon-containing molecules or compounds. So all of the biomolecules we're studying today are actually considered organic compounds, or organic molecules. Again, since they contain carbon as a major element in their composition. So looking at these chemical groups, we actually have some that we already talked about before. So here we have a hydroxyl group, so these are OH. This is very common in things like alcohol. For the rest of this lecture, we're going to talk mainly about carboxyl, amino, and phosphate groups. So these are three important groups that we're going to see in a bunch of different macromolecules or biomolecules we're studying in this lecture. Carbonyl groups are also very important, as well as methyl groups. You'll see those come up a little bit in this lecture as well. So for these chemical groups, you don't really need to memorize these. I'll never test you on these on exam or anything like that, so this is mostly just for you to be aware of them. When you're actually memorizing the structure of different types of macromolecules, knowing these different chemical groups is going to be very helpful. So definitely refer back to this table in your book, table 3.2, or you can look at this slide in the future when you're memorizing macromolecules. So the first macromolecule or biomolecule we're going to talk about are carbohydrates. So I like to think of this as being sugars. So in the picture on the left, I have a table sugar in the bowl, and then cubed sugar as well. So carbohydrates are made up of monosaccharides. So mono, again, means one, and saccharide means sugar. And there are many different types of monosaccharides. So some you may be familiar with are things like glucose and fructose. So fructose, of course, is the common sugar that's found in soda and soft drinks. So these are two common monosaccharides. And going back to some of the terminology we used earlier, each of these monosaccharides is considered a monomer. So monomer is a little bit more generic and less specific than saying a monosaccharide. When you say monosaccharide, you mean one sugar molecule. When you just say monomer, you mean one of something. So it's like saying I have one writing utensil versus one pencil. So pencil, of course, is more specific than writing utensil. So I can say I have two writing utensils, or I can say I have two pencils, two pens, one pencil, one pen. So you can think of monosaccharide being more specific and monomer being less specific. So the major function of carbohydrates is energy. So you can think of eating candy or something like that. You get a sugar rush, so your body's using that carbohydrates as energy. For plants, though, it can also be used as structure. So we'll talk a little bit about cellulose. So this is actually the component that's found on the outside of the plant cell, and this gives plants their structure. We also know cellulose as being dietary fiber. So now we're going to look more closely at each of those monomers or monosaccharides we were talking about. Specifically, we're going to look at glucose in this picture. So these are what are called the linear form of a monosaccharide. In most chemistry and biology textbooks, you'll see the ring form. So just as these names allude to, the linear form is a straight line, the ring form is a ring. So here's the ring form, glucose. So linear form and the ring form are pretty much exactly the same thing. Just a linear form you can imagine is wrapped around and connected, and the linear form is just a straight line. So here, if we actually look at these numbers right here, these numbers match up exactly with the carbons in the ring versus the linear form. So you can see how this linear form is wrapped around and created into a ring. I should stop here and mention the word carbohydrates. So a good way to see if something's a carbohydrate is to see if it has a lot of carbons and a lot of hydrogen. Oftentimes, you also see oxygen as well. So you're looking for lots of carbons, hydrogens, and oxygens. You can see lots of those right here, a lot of hydroxyl groups, a lot of hydrogens, a lot of carbon. So really the only macromolecule we're going to see in a ring form, again with lots of carbons and hydrogen and oxygen, are going to be carbohydrates. So if you see something in a ring form like this, oftentimes it's going to be a carbohydrate. So that's a good way to identify sugars or carbohydrates. So again, we have the linear form and the ring form. Both are essentially the same thing, just the ring form is a linear form wrapped around itself and connected. There are also multiple ways of writing the ring form. So we have what's called an abbreviated structure. So this abbreviated structure is not unique to carbohydrates. You often see this abbreviation in other biomolecules and macromolecules we're going to study in this lecture. But the way they've abbreviated things here is essentially each bend that you see is a carbon. So each of those circles I just made represents a carbon. So every time you see a bend in the line, that's a carbon molecule. Same thing with this simplified structure over here. Just they've removed the hydroxyl groups. So each bend is a carbon. So that's a carbon there, carbon there, carbon there, and carbon there. So whenever you see a bend in a line in any of the future biomolecules and macromolecules I'm going to show you, that means there's a carbon there, just they haven't bothered to write it out. So this is a shorthand way of writing out carbons. So going back to the naming scheme, just go over this again. One saccharide by itself is a monosaccharide. Two saccharides, we call that a disaccharide. So di means two. And then many saccharides linked together, that is polysaccharides. Again, you can use the more generic term polymer. But if you say polymer, we're not sure if you're talking about a protein or if you're talking about carbohydrates. So that can be anything in general. So more specifically, you should use the term polysaccharide so that we know you're talking about sugar molecules. So here's a question to answer on Blackboard. This is question number 2. How is the disaccharide below formed? So A, hydrolysis, or B, condensation? So this question is really just to check and see if you remember this idea of hydrolysis and condensation. And so hydrolysis, remember, hydro means water and lysis means to break. So this is breaking. Condensation, though, is building. So here, since we're going from monomers to a dimer, we're actually building. So the correct answer here is B. So this is a condensation or dehydration reaction, as your book puts it. And here is just the entire figure itself. So again, this is a dehydration reaction. So you're going from two monomers to what's called a dimer. And dimer, the di prefix means two. In this case, this dimer gets a special name. So a lot of disaccharides or dimers often get names. So two glucose molecules linked together is called maltose. So you may have heard of malt sugar. So that's made of lots and lots of maltose. Also, I should stop here and say that a common way to identify if something's a sugar is to look at this OS ending. So OSE usually means sugar. So glucose, maltose, those are both sugars. So we've talked about monosaccharides and disaccharides. Now we're going to talk about polysaccharides. Again, knowing your prefixes here is very helpful. So poly means many. So as opposed to one saccharide molecule or one sugar molecule or two sugar molecules, here we're looking at multiple sugar molecules linked together. And so there are 3 examples on the slide. We have starch, glycogen, and cellulose. So starch is probably pretty familiar to you. So starch is found a lot in potatoes. So French fries, potato chips, things like that have lots and lots of starch in them. And again, these are polysaccharides, so a bunch of sugar molecules linked together. So here we're looking at starch. You can see it has a sort of a helical shape. Starch is just a bunch of glucose molecules linked together. So again, we're looking at polysaccharides, long chains of sugar molecules linked together. Glycogen is probably less familiar to you. This is actually how your body stores sugar molecules. So here is actually an electron micrograph. So we're actually looking at a photograph of what these molecules look like. So each of these little dots is a bit of the glycogen chain. And again, I said glycogen is actually how your body stores glucose. So here we're actually linking together glucose molecules. You can see it's a little bit different. Instead of just a single helical chain like in starch, we actually have branching off from the chain itself. So polysaccharides can have many different structures. So they can be single helical structures like in starch, or they can have multiple branching chains like in glycogen. Lastly, we have cellulose. So this is actually how the plant itself gets structure. So we know it as dietary fiber. So this is indigestible plant material. And again, the outside of leaves, the stem, there's lots and lots of cellulose in plant material. And so cellulose is interesting because it's just a single long chain of glucose molecules linked together. What's special about it, though, is that it actually layers on top of itself. So you have layers and layers of cellulose, and you actually have hydrogen bonding in between those layers. So this is what makes actually glycogen very, very strong. You have this hydrogen bonding between these strains or chains of glucose molecules. So again, this is used to give the plant structure. So this hydrogen bonding is very important. So this is one of the reasons why the last lecture, chapter 2, modules 2.10 and 2.16 were so important, because we're going to see hydrogen bonding come up a lot in this lecture actually. So this is just one example where we see hydrogen bonding in cellulose, and that hydrogen bonding gives it extra strength. So cellulose is much stronger than glycogen or starch. So that completes carbohydrates or sugars. Next, we're going to move on to proteins, which are our second macromolecular biomolecule. So when I think of proteins, I like to think of whey protein. So if you're bodybuilding or you know any bodybuilder friends, they probably drink a lot of protein shakes. So our muscles are actually made up of lots and lots of protein, so they're trying to increase their protein intake in hopes that will be made into muscle tissue. So just like glucose and fructose were examples of monosaccharides or monomers, the name for a monomer of a protein is amino acid. So this is actually a very important word. So amino, if you break it down, actually means or relates to nitrogen. And of course, we've talked a little bit about acids in the last lecture. So here we have a nitrogen-containing compound that's also an acid. So in relationship to polysaccharides or monosaccharides or disaccharides, there really are only 20 amino acids. This is a lot different from sugar molecules. So there are lots and lots of different types of sugar. There's glucose, fructose, there's maltose. There are many, many different types of sugar molecules. But here, for the monomers of proteins, there are only 20 common amino acids. So if you take more advanced biochemistry or molecular biology, they may actually make you memorize all 20 amino acids. So for the polymer of amino acids, this is called a polypeptide. So a polypeptide is again just a bunch of amino acids linked together. So poly being the prefix here, poly meaning many, and peptide we'll actually talk about what that refers to in a later slide. So what exactly is a protein then? So when we have a polypeptide with 3D structure, we'll talk a little bit about how proteins actually form 3D structures later. But when it forms a 3D structure, we then start calling it a protein. So essentially, a protein is a polypeptide with 3D structure. In regards to the functions of proteins, so proteins are very important molecules in the body. So it can do lots and lots of different things. So to sort of take a reductionist approach here, I'm just going to say they're used to do work. Specifically, enzymes are the molecules that do work. So enzymes are proteins that perform lots and lots of work or reactions in the body. And when we start talking about cellular respiration and the mitochondria, we'll talk a little bit about enzymes and how they do what they do. Also importantly, proteins are used for structure. So of course you can think of muscles, you can think of keratin in the skin. So proteins are very important for giving structure to things. And just to make life easier for us, I'm going to put and many more at the very end of the function list. So proteins, again, are very important, diverse molecules in the body. So they can have lots of functions, so it's very hard to come up with a simple short list for us. But as we go through the semester, you'll find out that proteins have a lot of functions, and we'll stop and talk about each one as we go. So looking at the right-hand side of this picture, this PowerPoint slide, we actually have an amino acid. This is an amino acid right here. Again, I mentioned that when we say amino, we're actually talking about nitrogen. More specifically, though, we're talking about this amino group right here. So amino groups contain nitrogen and 2 hydrogen atoms. There's then a central carbon. So sort of gray sphere, that's the central carbon. We then have an R group. So the R group is called the variable group sometimes. So all amino acids have an amino group and a carboxyl group, as well as a central carbon and R group. What changes between the 20 different amino acids is the R group. So the amino group, central carbon, and carboxyl group are all constant. The only thing that changes is the R group or variable group. And lastly, as I said before, we have a carboxyl group. So this right here. And this is actually an acidic group. So this is acidic. So this actually raises the pH of solution. So this is why we call these amino acids. So they have this carboxyl group, which is acidic. It's actually a little bit more complex about that. If you take organic chemistry, you'll learn a little bit more about Zwitter ions and things like that. So here are the 20 common amino acids. So again, I should say these are common. So there are actually more amino acids, but these are the 20 that you'll commonly see in life. And here we have them grouped by our various properties. So many of them are nonpolar. So these are side chains that are nonpolar. So these are R groups that are nonpolar. And again, I said the carboxyl, central carbon, and amino group are constant. So here I can just circle all of them. So there is your amino group, central carbon, carboxyl, amino group, central carbon, carboxyl. Again, those are constant. You'll see them in all amino acids. So the only thing that's really changing for each of these amino acids is the side chain or R group or the variable region, or what have you. And that is in green in this picture right here. So for something like glycine, the R group is just a hydrogen. For something like alanine, it's a methyl group. Valine, you have this configuration right here, and so on and so on. But for all of these amino acids that are in green, these have 9 non-polar side chains. Next, we have polar side chains. So again, these are amino acids with polar side chains. So in purple, we have various amino acids with polar side chains. We also have electrically charged side chains. So these are things like aspartate and glutamate, and lysine, arginine, and histidine. You should also note that the electrically charged side chains can be acidic or basic. So in the next slide, I'll actually show you a little bit about what that means. But the main take-home message from this slide right here is that we have various different amino acids with various different properties. Specifically, nonpolar side chains, polar side chains, and electrically charged side chains, either being acidic or basic. So I don't want you to memorize these 20 amino acids. I just want you to appreciate the fact that we have different types of amino acids with different properties. So for the electrically charged side chains, I mentioned they can be acidic or basic. So here the definition we're using for acid is it gives off a hydrogen. So looking at aspartic acid, this hydrogen right here is donated to solution. And for glutamic acid, that hydrogen right there is donated. So remember, Acidic solutions generally have a high hydrogen ion concentration and a low OH concentration, whereas basic solutions have a low H plus concentration and a high OH concentration. So for their acidic amino acids, they're giving off this hydrogen and they're increasing the amount of hydrogen ions in solution. For basic side chains, these accept hydrogen ions. So right here, this is actually drawn after it's accepted. So here we had NH2 actually accept a hydrogen ion and it became NH3. Same thing for arginine. NH2 became NH3 when it accepted a hydrogen. And here this nitrogen became NH on histidine when it accepted a hydrogen. So again, it's accepting hydrogens actually from water. So we're going from H2O to OH minus. So that water gave these amino acids a hydrogen, and that converted H2O to OH, and that is increasing the OH concentration in our solution and making it more basic. So again, acids give off a hydrogen, bases accept a hydrogen. And that's exactly what we're seeing here with aspartic acid and glutamic acid giving a hydrogen, and lysine, arginine, and histidine accepting a hydrogen from water. So again, here we have the amino acid structure. We have the three common or three universal parts. We have the amino group, so NH2. We have the carboxyl group, or carboxylic acid, and then we have the central carbon. So the R groups can actually be hydrophobic and hydrophilic. So hydro means water. Phobic, you can think of a phobia. So you have a phobia of spiders, or you have a phobia of earthquakes. So phobic means scared of. For philic, that's actually the exact opposite of phobia. So that means loving. Hydro, again, means water. So some amino acids don't like water. So these are ones like leucine. And some do like water. So serine or aspartic acid. So those like water or are hydrophilic. So really, in general, the nonpolar amino acids don't like water. The hydrophilic amino acids are usually polar. And so I'm going to introduce you to a concept. Like interacts with like. This is a very important concept that we're going to come up with, or actually see again in later lectures. So like interacts with like. So since water is actually a polar molecule, water likes interacting with other polar things. Specifically, polar amino acids. So polar amino acids like interacting with other polar substances such as water, which makes them hydrophilic. Water, however, does not like interacting with nonpolar things. So nonpolar amino acids generally don't like interacting with water. Again, water is polar. Nonpolar amino acids are nonpolar, so they don't like interacting. This makes certain amino acids hydrophobic. So that's why you can make this generalization here. Again, nonpolar amino acids are hydrophobic, and polar amino acids are hydrophilic. So like interacts with like. In this case, water is polar, so polar amino acids like interacting with water. So here's a question to answer on Blackboard. So now I want you to be able to identify some of the macromolecules we've talked about. And specifically in this slide right here, I want you to identify which of these is an amino acid. So think back to the last few slides I showed you. What are the common characteristics of an amino acid? So what groups do we see in amino acids that make them special? So for this question, it's very helpful to remember the parts of an amino acid. So remember, you have the NH2 group, or the amino group. You have a central carbon. You have a carboxylic acid group. And you have your R group. So if you remember these 4 parts, it should be easy to identify which one of these is an amino acid. And in this case, C is the correct answer. So it's a little bit tricky of a question since I've actually flipped it on its side here. So whereas you saw it upright in most pictures, so like the one right here, this is upright, C is actually sideways. So here again you have the amino group, you have the carboxylic acid right here, carboxyl group, and then you have the R group right here. So again, C is an amino acid. If you remember the 4 parts of amino acids, it should be easy to identify which one of these is the correct answer. For A, B, and D, those are all polysaccharides, not polysaccharides, monosaccharides and disaccharides. And so here I believe we actually have glucose. So remember, saccharides can actually come in these ring forms. So here we have the characteristic ring. Here we have a disaccharide. So these are 2 rings linked together. And also remember, we can have a linear form as well. So there's the ring form and linear form of carbohydrates. So this is the linear form right here. So for each of these, we don't really see the characteristic groups. So we don't see the carboxyl group, we don't see the amino group in many of these. We also, of course, don't see the R group as well. So this leaves us with C as being the best answer here. And again, we see the amino group, the carboxyl group, the central carbon right there, as well as the R group. So C is the correct answer here. So I introduced this word polypeptides. So we talked about the poly part. That's straightforward. That means many. Now we're going to talk about the peptide suffix here. And to figure out what this means, we're going to draw the bond formed or implied about the reaction below. So here again we have a dehydration reaction. So OH plus this H right here is going to produce water. And when that happens, we're going to have these 2 amino acids linked together, and we're going to have some special bond formed, which we're going to talk about later. So let's actually draw this. So here we have our carboxyl group. That OH and H, remember, is going to be removed. So now we're going to have a bond between the C and the N. We still have 1 H left. So this H right here is still left. Only one of the H's or hydrogens are donated to form water. So here we have the C double bond O and NH. Then we have the central carbon, H over here, R group right there, carboxylic acid again, carboxyl group, central carbon right there, H, R, and then amino group. So here is our final drawing. So this bond right here between our carboxyl and amino, or what were the carboxyl amino groups, is actually called a peptide bond. So this is how we get the word polypeptide. So poly means many, and peptide refers to the peptide bond linking these amino acids together. And so here's that, just written out in a nice figure. So again, we have a dehydration or condensation reaction. That removes water. And what's left is this peptide bond. So C double bond O, and then NH to the right of that. So that is now a dipeptide. Again, di meaning 2. So we formed a dipeptide by bonding together 2 amino acids. So we've talked about polypeptides, but now let's talk about proteins. So polypeptides, when you get a 3D shape from those polypeptides, we then call that a protein. This forming of the 3D shape is often called folding. So that's the technical term for it. So when a polypeptide folds or forms a 3D shape, we start calling a polypeptide now a protein. And so there are 4 major levels or structures of proteins. So the primary structure sometimes written like this, is the amino acid sequence. So it's just valine, proline, arginine. So it's just the sequence of amino acids. Secondary structure is when hydrogen bonding starts coming into play. So again, hydrogen bonds are very important in a lot of different macromolecules, including proteins. So we have alpha helixes and beta pleated sheets formed during secondary structure, and this is due to hydrogen bonding. Once we reach the level of tertiary structure, this is now 3D structure, and now we have a true protein at this level. So once a polypeptide has reached tertiary structure, it's now become a protein. So now, in addition to hydrogen bonding, we have ionic and hydrophobic interactions. So this is now 3 different types of bonds, each playing a role to give that protein a 3D structure. And then quaternary is the last type of structure in proteins. So this is multiple polypeptides linked together. And specifically, these are linked by covalent bonds. So not by hydrogen ionic bonds, but specifically by covalent bonds. So let's actually look at a picture of what these 4 structures look like. So again, at the primary level, and for now I'll write out the shorthand way of writing all this. So 1 with the circle next to it means primary, 2 with the circle means secondary, 3 with the circle means tertiary, 4 with the circle means quaternary. So at the primary level, again, it's just a sequence of amino acids. So here we just have a bunch of amino acids linked together, and this is the primary structure of the protein. Once we get to the secondary level, we start having hydrogen bonds coming into play. So we get these beta pleated sheets, which looks like folded pieces of paper, and we get these nice little helixes called alpha helixes. So this is a secondary level of protein structure. On the third level, we finally get that 3D shape. So now things are coming together. You can see it almost looks like a ribbon all tangled up. So now you can start seeing the 3D structure of the protein. In the last and quaternary structure, we have multiple polypeptides linked together. So here are 1 and 2 polypeptides, 1 in light blue and 1 in dark blue. So these are the 4 levels of protein structure. So please do memorize for exams. For some reason, students seem to do poorly on questions related to these structures. So remember, there are 4 structures of proteins. Primary is just the amino acid sequence. Secondary are beta pleated sheets and alpha helixes due to hydrogen bonding. Tertiary structures when you actually get the 3D shape of the protein. That's due to hydrogen ionic bonds and hydrophobic interactions. And then quaternary structures where you have covalently bonded polypeptides linked together. So please do remember that there are 4 different structures of proteins, and also remember the properties of each structure. So here's the 4th question to answer on Blackboard. So tell me, just your best guess, how many different proteins do you think there are in the human body? So here the correct answer is actually D. There's actually 100,000 or so different proteins in the human body. It's important to remember here that proteins do a lot of different things. So think about, what do proteins do and what is different about each protein. So as you can imagine, proteins have a lot of different functions, so we need a lot of different proteins. But also there are 20 different amino acids, so we can have many, many different configurations here. So based on those configurations, they get different 3D shapes. And those 3D shapes confer some sort of function or purpose. So here this question is just sort of pointing out that proteins are extremely diverse molecules. So they have diverse functions, diverse structures. And as we go along the semester, again, we'll be going over some of enzymes and proteins that we find. So first we talked about carbohydrates, then we talked about proteins, and now our third biomolecule or macromolecule is going to be nucleic acids. So the most common nucleic acid you've probably heard of is DNA, and that's exactly what I have shown in this figure. So here we have 2 strands of DNA intertwined together to give it its common double helical shape that you're probably very familiar with. So nucleic acids are actually very important in the cell, so it actually codes for amino acids. In other words, it actually tells the cell what proteins it needs to make. So DNA contains all the information to make proteins. So that's one of its most important functions, coding for proteins. The monomer of nucleic acids is called nucleotides. So you may actually hear me go between the two, so sometimes I'll say nucleic acids, sometimes I'll say nucleotides. But remember that nucleotides, more specifically, is the monomer of nucleic acids. The polymer, though, so linking together of many nucleotides, is called deoxyribonucleic acid, or DNA, and ribonucleic acid. Sometimes you'll also see polynucleotide. Polynucleotide is less common of a phrase. You probably won't see it very often, but know that polynucleotide refers either to DNA or RNA, so those are the colloquial terms that we use. And the function, again, is information storage and transmission. So you know DNA as being the heredity molecule, so it's genetic information for all of our cells. And again, it contains all the information needed to code for amino acids and proteins. So here's the basic structure of a nucleotide. So there are three common parts. There's the phosphate group. You can see the central phosphorus right there. There's a sugar. And then there's a nitrogenous base. So three major parts, again, are phosphate group, sugar, and nitrogenous base. So the sugar is actually what changes between DNA and RNA. So DNA equals deoxyribose. Again, remember sugars have that O ending, so ribose is the sugar. RNA is just ribose. And don't worry too much about this for now. We'll go into much more detail in later chapters of the textbook. And then lastly, we have a nitrogenous base. As we're going to see, the nitrogenous base is where we get most of the variety. So for DNA, there are four major nitrogenous bases. There's adenine, or A, guanine, or G, cytosine, or C, and thymine, or T. So essentially, for DNA, the sugar and phosphate group is going to be constant, and the nitrogenous base is going to be what changes. So essentially, depending on the sequence of nitrogenous bases, that's going to produce a different protein. And again, we're not going to go into too much detail about DNA and RNA for now. In later chapters, we'll go into much more detail. DNA and RNA can also form helixes. So as you're probably familiar with, DNA has a double helix. So here's one of the helixes, and here's the second. So those helixes are linked together to form a double helix. So here's question 5 for Blackboard. So which of these is a nucleotide? So now that you've taken a chance to answer that question, let's go over the answer. So again, you want to remember the three parts of a nucleotide. So there's the phosphorus group, there's the sugar, and then you have the nitrogenous base. So essentially, nucleotides have three major parts. So A, if you look at that, that's actually an amino acid. So we have that amino group, and we have the carboxyl group, and the R group. B is a sugar, so it's not the correct answer. And C is also a sugar, just in its linear form, so not in the characteristic ring form. So that leaves us with D being the correct answer. Again, we have the phosphate group, the sugar, and the nitrogenous base right here. So D is the correct answer here. And again, a lot of these questions you're going to see on the exam, so I'm going to show you some sort of molecule, and I'm going to have you identify which of the major macromolecules it falls under. So it's very important that you're able to do this by the time the exam comes around. So the final biomolecule or macromolecule we're going to talk about are lipids. So when I think of lipids, I think of things like butter. That's a very common food substance that has lots and lots of lipids in it. So lipids are actually very hard to define as a group. One common characteristic of those is that they're all hydrophobic. So you can think of oil, something that's also made up of lots and lots of lipids. That does not mix well with water. So if you mix it with water, it forms two layers, and that's because lipids are hydrophobic, so they dislike water. Again, lipids are very hard to define, partly because they lack any true monomer or polymer. So there's no common characteristic or structure I can show you to help you identify lipids. However, you can sort of break down lipids into three major groups. So there are triglycerides, also known as fats. So that's the common word we use. There are phospholipids, and then there are steroids, which you're probably also familiar with. So these are things like sterols and cholesterols. Those are also names you can use to reference steroids. In regards to the function of each of these, for phospholipids, those are very important for forming the cell membrane we talked about on the first day. So those form the cell membrane. Triglycerides, so you can think about body fat, so that's used for energy, and specifically energy storage. So not for a quick burst of energy like carbohydrates, but for long-term storage of energy. And then lastly, we have cholesterols or steroids. So these are used for signaling. You can think of hormones as being a very good example of a steroid, and also many more functions beyond that, some of which we'll talk about in later lectures. So here's a triglyceride. Triglyceride means, or is commonly referred to as fats. So that's the common word we use. There are really two major parts of a triglyceride or fat molecule. There's a glycerol group. So this combination of carbons linked together with these oxygens right here, that's a glycerol group. And then you have three fatty acid chains linked together. So each of these fatty acid chains is sometimes called a tail. So essentially, a triglyceride has three tails. And that's sort of how it gets its name. So the tri means the tails, and the glyceride refers to this glycerol group right here. So that's how we get the name triglyceride, commonly referred to as fats. And so here are some common foods with lots of triglycerides. So things like meat, butter, have a lot of saturated fats. This term saturated we'll talk more about later in a future lecture. Things like olive oil and salmon also have lots of fats, specifically unsaturated fats. Again, we'll talk about saturated versus unsaturated fats in a future lecture. Next up, we have phospholipids. And here I have the balloon that we talked about on the first day. So you can think of the balloon itself being the cell membrane of the cell. And again, that cell membrane is made up of phospholipids mainly. So there are really two parts of a phospholipid. There's the hydrophilic head and the hydrophobic tail. So remember, hydrophilic means water-loving, and hydrophobic means water-fearing. So the hydrophilic head is made of a couple parts that we've heard about before. So just like in triglycerides, we have a glycerol group, and we also have a phosphate group. So of course, you have the prefix phospho, so that relates to the phosphate group. The lipid group actually refers to the fatty acid tails we have here. As opposed to triglycerides having three fatty acid tails, remember tri, three, here phospholipids only have two fatty acid tails. And the more technical term for tails is chains. So the actual fatty acid tails themselves are hydrophobic. So they have lots and lots of nonpolar bonds between each of those carbon and hydrogen. If you have questions about polar versus nonpolar, I'd recommend going back to the Chapter 2 lectures where I discuss the difference between polar versus nonpolar bonds. So again, lots of nonpolar bonds here. Because there are so many nonpolar bonds, that makes part of the molecule hydrophobic. However, there are a lot of polar bonds in the hydrophilic head. So the head, since there are so many polar bonds, so for instance between the carbon and the oxygen, both of which you see in the glycerol, in between the phosphorus and the oxygen, also polar bonds. So you have lots of polar bonds here. So this makes the head of the molecule, or the head of the phospholipid, hydrophilic. So we have an interesting example of a molecule that both loves water and hates water. So the head is hydrophilic and the tails are hydrophobic. So we have a special word for this. We call this amphipathic. So this is a very important word, so make sure you memorize it. So amphipathic means both it loves water and it hates water at the same time. So a phospholipid is a very unique example of an amphipathic molecule. And why this is important, we'll talk about later in the lectures. When we talk about actually the cell membrane itself, we'll talk about how phospholipids play a role in forming the cell membrane and why amphipathic molecules are important in doing that. And here's that just written out. So remember, hydrophobic means fear of water. So that's a lot of nonpolar covalent bonds. You can think of oils and fats. Hydrophilic things love water, so there are lots of polar covalent bonds. So things like protein powder dissolve very well in water, so they're hydrophilic molecules. And then lastly, we have amphipathic molecules, so they both love and hate water at the same time. Again, the head of the phospholipid, which is made of glycerol and phosphate groups, are hydrophilic, and the fatty acid tails are hydrophobic. So phospholipids are a good example of an amphipathic molecule. Lastly, our third and final major group of lipids are cholesterols, or as your book refers to them, steroids. So they have a common four-ring structure. I like to refer to this as sort of the Tetris shape you see. So you play lots of Tetris, you're probably familiar with that shape. So it has the same exact shape like this. So it has four rings linked together. And just like all the other lipid molecules, it's nonpolar. So these do not dissolve in water and they're hydrophobic. So two common steroids you probably heard about are testosterone and estradiol. So these are very important molecules for sex determination. Of course, testosterone you find much more in males, and estradiol you find much more in females. So if you go into physiology, or if you take more advanced biochemistry, you'll learn more about how these molecules actually play a role in development and why they're so important. But for now, just know that testosterone and estradiol are good examples of steroids. This is a table from your textbook, and I think it's a very good table to go over. So here we're actually going to go over the four major groups of biomolecules. And again, remember the point of this lecture is to sort of organize ideas. So hopefully I presented the material in such a way where you can actually organize the information. So first we're going to talk about carbohydrates. So the functions are energy for cell, raw material, plant cell support. And so another function of carbohydrates is energy storage. So you can think of starches and glycogen for humans. So that's all about storing energy. So if you ate raw glycogen, that wouldn't give you a sugar rush like eating or drinking a soda would. So carbohydrates can be used for quick energy and they can be used for energy storage as well as plant cell support. So for energy for cell or raw material, you can think of things like glucose. Again, starch and glycogen are good examples of energy storage. And then for plant cell support, remember we have cellulose. Cellulose had all those hydrogen bonds That gave cellulose much more structure and support compared to other polysaccharides. Next up, we have lipids. Again, lipids don't form polymers. There's no true monomer of a lipid molecule. So first up, we have the function of energy storage. And so what's a good example of energy storage? That's, of course, fats. Or the scientific term is triglycerides. So you can think of body fat. So it's all about storing energy. Next up, they give us the example of phospholipids. So remember, phospholipids are very important for the cell membrane. And lastly, we have hormones. So those are the steroids. So steroids, remember again, are things like testosterone and estradiol. Okay, going down to the next row, we have proteins. So here they give us 3 blanks. So here we're going to label the actual names of the different parts of an amino acid. So remember, we have the amino group right here. So G is amino group. We'll abbreviate amino here. We have I, which is the R group, also sometimes known as a variable region or variable group. And lastly, H is the carboxyl group. So remember, there are really 3 or 4 major parts of an amino acid. There's the amino group, the R group, and the carboxyl group. And here they haven't labeled it, but there's also the central carbon as well. And here they have a bunch of different examples of proteins. So something like lactase, that's for breaking down lactose. So lactose is, of course, the sugar you find in milk. So here we can say J would be something like digestion. Hair and tendons, so that's more about support. Muscle proteins, say movement. For something like transport, we'll talk more about this later when we get to diffusion or osmosis. But some proteins can actually act as pumps. So they can actually move things across the cell membrane. Next up, you have communication. So they've already done this for us. This is signal proteins. We have antibodies. This is all about immune system. And they've labeled the last 2 or so examples for us. So here you can see that proteins have many different functions. And again, they are responsible for doing a lot of work in the cell. So as we go through the course again, we'll go over each of these examples and talk a little bit more about how proteins play a role in each of these. Okay, so the last row here we have is nucleic acids. Again, just like proteins, it has 3 blanks for us to label the parts of a nucleotide. So O, remember there's a phosphate group. We have a sugar. And then lastly, we have a nitrogenous base. So those are the 3 parts of a nucleotide: the phosphate group, the sugar, and the nitrogenous base. So for our functions, we have heredity. So as I said before, DNA is all about storing information or storing heredity information. Here it's not very clear what they want for the function here, but it may be that they want protein coding. So remember, DNA and RNA are very important for coding for proteins. So what actually determines the amino acid sequence in a protein is the DNA or RNA sequence. So I hope that this lecture has helped you organize some of the information in the textbook. Again, if you have any questions, please do feel free to contact me at my email address. So in the next lecture, we're actually going to talk about the cell itself. So we're going to talk about organelles. Specifically, we're going to talk about things like these lipids right here, how they play a role in the cell. We're going to talk about carbohydrates, proteins, and nucleic acids. So we're going to talk about how each of these biomolecules or macromolecules play a role in the cell in the next chapter.